Whenever you're ready.
My opponents has presented some negative sides of animal testing, but I'm going to prove you that these are not correct. The first point is that animal testing is essential for safety in humans. So point A is animal testing can have potential effects to animals, but it's necessary for disease control and ensure food safety in humans. According to Dr. Scott, millions of people all over the world are alive today thanks to animal research. Much of this knowledge has also been used to tackle diseases that affect animals themselves, including cancer. Also, according to Dr. Natsu, the different drivers of using a vaccine in livestock has a positive effect on examining its safety and efficacy in a large number of herds, which is safe for human consumption. Therefore, animal testing is beneficial for food consumption and surgical operations. My subpoint B is that animal testing requires less time and exhibits less harm than human testing. According to Dr. Stadler, there is also a dark side to human experimentation, a long history of dangerous and harmful experiments performed on non-consenting patients. If animal testing is cruel, then human testings can never be seen as a testing method. My subpoint C is that animal testing are highly reliable to predict results of different diseases in humans. According to Dr. Manjipudi, non-clinical testing in animals is an important and critical component of risk assessment paradigm in developing new drugs. The basic assumption was that if animals used in laboratories experimentally contracted an infection and were cured, there was a high probability of stopping the same disease in humans. As originally conceived to be a valid model of human health concerns, the animal disease must have the same biological mechanisms, symptoms, and responses to treatment as the theoretically similar human counterpart. So we can see that animals and humans share many similarities. My point 2 is that many organizations and laws in the United States are monitoring the public health service policy on humane care and use of laboratory animals. My subpoint A is that animal research have not included species such as rats, mice, and birds to be protected under the Animal Welfare Act. According to National Research Council Committee, despite numerous efforts by the animal protection community to change the AWA to include rats, mice, and birds, an amendment was recently passed by Congress to permanently exclude rats, mice, and birds used in research for coverage by the Animal Welfare Act. Therefore, it's not like what my opponent said, that lab animals are poorly documented. My subpoint B is that the current animal research protocol is well justified by the Animal Welfare Act. According to National Research Council Committee, the act, now known as the Animal Welfare Act, has been amended 4 times, each time elevating the standard of animal care. First, an Animal Welfare Information Center was established to provide researchers with a database of alternatives to painful animal experiments. Second, each research facility in the United States using protected species must register with the USDA and establish an institutional animal care and use committee to review all experimental protocols involving live, warm-blooded animals. Therefore, painful animal experiments are highly regulated by the AWA and IACUC. My third point is that the use of alternative test methods, such as the use of human cells, are economically costly to develop and is not yet proven to be useful or safe. My subpoint B.A. is that cell or tissue culturing cannot yet replace animal testing. According to CFHS, the National Animal Welfare Conference, however, using cells and tissue culture does not allow researchers to see the effect of substance in living body with all its complexities. This method is valuable for research and can help limit the use of animals, but it cannot replace them entirely. Currently, the alternatives are still being developed and cannot yet replace animal testing. My subpoint B is that model animals share many similar genes as humans for testing. According to Dr. Ken Hinch, in an attempt to overcome the limitations of animal model, researchers are genetically engineering animals by removing or adding genes that they believe relate to specific human diseases. The underlying assumption here is that these newly genetically constructed animals will be more human-like. Since lab animals have much shorter lifespan, testing on these human-like animals will make it much easier and convenient to manipulate the tests. My subpoint C is that further studies on the alternative testing method is necessary to provide sufficient results. According to Dr. Dobchova, it's still the only general accepted testing for assessing the carcinogenicity of chemicals. Over time, different alternative approaches have been developed with the final goal to replace the bioassay. Therefore, none of these strategies alone provide sufficient assurance of the accurate predictions.